Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts of biosciences. Today we are going to discuss about C2 photosynthesis. The C2 photosynthesis is also known by another name, that is glycine shuttle. In this video we will look at the pathway, we will look at the significance of this pathway. We are aware that the fixation of carbon is carried out by an enzyme known as Rubisco. Rubisco has got two enzymatic activities. One is carboxylation reaction, which we have elaborately discussed in a video on the Calvin-Benson cycle. And the second enzymatic activity is an oxygenase activity. The carboxylation reaction fixes a CO2 molecule to ribulose 1,5-bisphosphate, and we will be getting two molecules of a 3-carbon compound, that is 3-phosphoglycerate. Whereas oxygenation reaction will result into fixation of an oxygen and results into 1 3-carbon unit and 1 2-carbon unit. This will reduce the fixation of carbon dioxide or photosynthesis into about 25 to 40% reduction. Economically, it is not a process that is beneficial to plants. A plant has to invest lot more of ATP molecules in order to fix back the 2-carbon unit into the 3-carbon unit. Rubisco, as we know, is a very large enzyme. It has got large subunit which are coded by the large subunit coded in the chloroplast. It has got small subunit which are coded in the nucleus. These are the small subunit, these are coded in the nucleus, and then this arranges itself into a very large enzyme. Now this enzyme has been probably evolved maybe about 3 billions of years ago. We know that 3 billions of years ago the carbon dioxide concentration was very high in the atmosphere. When the CO2 level was very high, probably Rubisco opted for the carboxylase reaction. Over a period of time, when oxygen concentration began increasing in the atmosphere to the level at which we have now, which is about 21% is oxygen in the atmosphere, probably it has evolved in the oxygenase activity as well. We know that the oxygenation reaction is a side reaction which is not a preferred reaction for a plant. It's a waste of energy. In this context, we may begin to wonder why this oxygenation reaction was not switched off in the last, let's say, in the 3 billions of years. And also, Rubisco is a very slow enzyme. The catalytic activity of this enzyme is also very slow. Probably it is fixing about 2 to 3 CO2 molecules, 2 to 3 per second. So it is a slow enzyme. We naturally begin to wonder, if a plant has to synthesize a lot of starch or photosynthesis has to occur, why the catalytic rate of the enzyme was not improved over a period of time. This is something mysterious. Scientists have been working on this enzyme. So they have tried several things in order to increase the efficiency of this enzyme. What they have done is they have first of all tried to knock out the oxygenase capacity. So make it oxygenase minus, but it was not successful. Why it was not successful? We know that in the carboxylase as well as in the oxygenase capacity is at the same active site. So therefore, knocking off this activity means it is also we are knocking out the carboxylation activity. So therefore, it was not successful. Then scientists tried to genetically engineer Rubisco with greater catalytic activity. So they tried to increase the K-cat, but that was also not very successful. Now, in the alternate mechanism, another mechanism which they are trying to improve greater photosynthesis is they are looking at converting the C4 crop plants into this activity. They are trying to improve in the C3 plants. So this is the way in which the current research is going on with regard to improving the carbon fixation in plants. At this stage, we need to understand what determines the ratio of carboxylation to oxygenation in Rubisco. There are several factors. The first one is the catalytic rate of Rubisco enzyme. We have seen that the catalytic rate is very slow for the Rubisco enzyme. And the second factor is in the preference of Rubisco for the two substrates. The two substrates are carbon dioxide and oxygen. In order to understand this concept, we need to look at three facts. The first fact is a concept called K-cat. K-cat means the turnover number. What is the meaning of the turnover number? If the turnover number means the number of molecules of substrate that can be handled by the enzyme at saturating concentration. That is the turnover number. A second concept that we need to understand is a concept of Km value. We know that from our enzymology classes, this is the Michaelis-Menten constant. Km means that the concentration of the substrate at which the enzyme is showing half the maximum velocity at saturating concentration. So the Km value will indicate about the activity of the enzyme. The third factor is known as the specificity factor. Specificity factor means the preference of the enzyme towards the two substrates, carbon dioxide and oxygen. This can be easily calculated by taking the ratio of K-cat to Km for each of the substrate. This can be written as follows. So therefore specificity factor is equal to the K-cat for CO2 divided by the Km for CO2 divided by the K-cat for the second substrate, that is O2, divided by the Km for O2. This ratio is this is the specificity factor. Generally, for large plants, the specificity factor is roughly about 80 upward. So, which means the numerator has a significant role in deciding on this specificity factor. When we discuss about a specificity factor, we should also remember that if the concentration of oxygen in the atmosphere is about 21% and CO2 is only about 0.035%. So plenty of oxygen is in fact available. Now, when it comes to a plant, let us look at the cytoplasm as well as in the stromal conditions. What will happen is, if the solubility also plays a key role. Now, we know that when the temperature increases, the solubility of CO2 is decreasing, whereas it has no effect on the solubility of oxygen. which means that all these are contributing towards CO2 making it less available when the water content is low or when the temperature is high. So therefore, when the water content decreases as well as when temperature increases, the availability of CO2 is less and oxygen is more for the enzyme to fix, and which lead towards photorespiration.